into detail regarding the actual experiments I've done, I'd like to give you all a crash course in genetics. For some of you it's going to be a refresher. For some of you it might be an introduction, but I hope it will help put my project into a bit better context. And hopefully by the end of this you'll feel as I do about how important genetics is as an area of study. If nothing else, you'll appreciate the new exercise. Now, my colleagues have already spoken about prokaryotic cells. What I'd like to show you here is a typical eukaryotic cell. If you were to swab the inside of your mouth now, then place it under a microscope, this is what you would expect to see, although possibly not with such vivid colours. And you can see a few of the different features that you'll see inside the eukaryotic cell. These are membrane-bound organelles, and that's what actually makes it a eukaryotic cell. What I'd like to bring your attention to is the large red circle just off centre there. That's the nucleus of the cell. And inside that nucleus there's a fluid called chromatin, and inside of that chromatin is where you'll find the DNA. And the DNA will be floating around in long strands, which in that state are not actually visible save for under extreme magnification. However, prior to cell division, mitosis or meiosis, what you'll find is that the DNA will actually compress itself into the chromosome. You can see one just on the side there, an example of how the DNA is actually structured there. Now, what I actually have is a three-dimensional model here of a sample of the DNA strand that you can see just on the side there. Now, this is only a section of it. In reality, each one of these nucleotide base pairs would extend for millions of base pairs in that direction and millions in that direction. These molecules are extremely complex, and they're used for the storing of information. They're essentially the blueprint for all life on Earth. Now, DNA itself, these long strands, they're only made up of 4 different compounds, and I actually count them here. Now, what we've got is adenine, thymine, guanine, and cytosine. And on these models here, I hope you can all see them, I know some of you are a bit far back. You've got carbon, nitrogen, hydrogen, and oxygen. And each of these spheres actually represents just one of those atoms. So I hope that will give you some sense of just the kind of scale we're actually talking about here. It's really, really tiny. Now, all life on Earth, whether it's simple bacteria all the way up to much more complex organisms, including ourselves, is actually just, our DNA is just a remnant of these 4 nitrogenous bases attached to a sugar phosphate backbone. And the sugar phosphate backbone is actually the outer layer that you can see running around the outside of the DNA here, which kind of holds the structure together from the outside. Now, these nucleotides, all the DNA, are arranged, as you can see, they're like rungs in a ladder, and every 3 of these can be equivalent to what's called a codon. And when you have these codons, will each code for a specific amino acid, and when you have a chain of codons that code for a specific sequence of amino acids, otherwise known as a protein, that's what a gene is. That's what we mean when we actually talk about a gene. And these genes can control a number of different things. I mean, they actually affect everything. There's only a small number of them we can observe. And if you look around the room, you can see people have different hair colours, they have different eye colours, they have different heights. And all of these things are controlled by different genes. So even though we've got the same overall structure, there's a lot of difference. And these observable differences are what we would refer to as our phenotypes. Now, when an organism produces gametes, and these are sex cells, it does so through a process called meiosis. And you can see some of the first stages of meiosis here. Now, this is just a theoretical organism. I've represented the chromosomes here. There's 2 pairs of chromosomes, and I don't know if you can make out the colours. The blue ones would be paternal, and the red ones would be maternal, showing the red ones have come from the organism's mother, the blue ones have come from the organism's father. And this is the set of DNA in this theoretical organism has 2 pairs of chromosomes. In humans, you would actually see 23 pairs of chromosomes, but I didn't have enough data to do that. It would be a ridiculous image. So what you can see here is, in the first image, you've got the outline of the nucleus, it's breaking down, and the nucleolus, which is the small ball of planar there, disappears, and that releases all the histones and the protein scaffold, which let the DNA wind itself up into these chromosomes, which are the X-shaped pieces of planar that you see there. And then in stage 2, and it's not obvious because you can't make out the colours, but some of the genes have actually switched from the blue to the red. So the red chromosome is now, half of the red chromosome is now carrying 2 blue genes, and half of the blue chromosomes are now carrying some red genes. So this is called chiasmata. And it's important to note that when genes switch like this, they're always switching their corresponding genes. So if the eye colour gene were to move from one of the blue chromosomes to the red, then the corresponding eye colour gene would move from the red chromosome to the blue. So there's still a full set of DNA there, it just gets shuffled around. These chromosomes will separate, and you can see how the cell divides into 2 cells, and then the second phase of meiosis begins, where the chromosomes are actually pulled apart, and these chromosomes are actually 2 sets of wound DNA that are identical. Beforehand they're identical, but because of chiasmata they're now non-identical. So they're pulled apart, and the cell separates into 4 distinct cells. You can see 4 nucleuses forming here, each one of which contains half of the information needed to build a new organism. And this is why gametes have half of the information that are needed, so that when a new organism is made, it has 100% of the DNA that it needs. And it's because of this division that we can actually draw a Punnett square, like the ones that you see here. And these are Punnett squares for monohybrid inheritance. Now I've used the letter R here just to represent a general variable. These can represent any gene that you want to track in monohybrid inheritance. And in general, the capital R's refer to a dominant gene, and the lowercase r's refer to a recessive gene. And you can actually see along the top of the squares the contribution from the father, and on the side of the square is the contribution from the mother. So the first Punnett square, the one on the left there, you've got what are known as heterozygous genotypes in the parents. They have 1 dominant and 1 recessive gene. And the 4 boxes represent the 4 possible offspring they can have, or 4 characteristics their offspring can have of these genes. So you can see there's the homozygous dominant, which is the top left-hand corner, and the bottom right-hand corner is homozygous recessive. This means they have the same version of the genes, the same dominant or recessive genes. And homozygous dominant will present the dominant phenotype. Heterozygous dominant will present the homozygous recessive will present the recessive phenotype, and the 2 heterozygous versions, capital R, lowercase r, will present the dominant type, because the dominant gene will mask the recessive gene. And then I've included the second Punnett square here, so I've just changed the characteristics of the father. It's got 2 capital R's to illustrate that now the properties of any offspring alter. There are no homozygous recessive combinations in this second Punnett square, so any children produced in this coupling, any offspring would have the dominant phenotype. Now, these are the kind of Punnett squares that were drawn from the research of Gregor Mendel back in 1865. He was studying pea plants, and at the time he didn't know anything about chromosomes or genes or phenotypes. None of these kind of words really existed, but he still found the same ratios that you would expect to find. So it would be in the top left Punnett square there, a 3-to-1 ratio of dominant to recessive phenotype. Now, when he did his research, all of the pea plants he used actually, just by pure luck, had monohybrid inheritance. All the genes were not linked to one another, they could all be inherited separately. So he drew from this research 3 principles of inheritance. The first one is the law of segregation, which is that every parent has a pair of alleles for every gene and will pass only one onto their offspring. And that's what I mentioned before in meiosis, how only half the information is passed on, but you've got half the information you need for every characteristic. The third law, and I'm skipping one, so I'll come back to the second one, is the law of dominance. And this is that recessive characteristics will always be masked by dominant characteristics if the gene for both is present. That's what you see in the heterozygous pairings, even though they're capital R, lowercase r, the capital R phenotype is the one that presents. And the second law is the law of independent assortment. And this states that genes of separate traits are passed on independently of one another. And this actually brings me to dihybrid inheritance, because in fact we know this law is not universal. There are some genes which are linked to one another. And in this example, this particular table you're looking at here, B and G, they're the characteristics for vestigial and wild-type wings on fruit flies, Drosophila melanogaster. And these genes are actually linked to one another, so they are not separated during chiasmata. They do affect one another, so they're not passed on according to the second law of inheritance. And you can see it leads to a much more complicated Punnett square there. And this actually brings me to my research question. Does Mendelian inheritance provide a suitable model for accurately predicting dihybrid inheritance in Drosophila melanogaster? And the reason I'm asking this question is because Mendel's original research was on monohybrid inheritance, even though he may not have been aware of it at the time. So I'm looking at dihybrid inheritance, which is what affects many of the characteristics of Drosophila melanogaster, to actually see if the ratios that Mendelian inheritance says we should get actually do not true. So I did this by approaching the college's prep group, who were kind enough to provide me with some Drosophila. And you can see some pictures here of the general tools I use. It's actually quite a simple experiment. It's a bit time consuming, but there's no massive pieces of technology that you use for it. So the first thing I had to do was take the Drosophila that the lab provided me with, separate them out, and then etherize them. And I did this so that I could examine them, because obviously they move around quite a bit, and they're very, very small, so you need them to be unconscious. And I separated them, and I recorded their characteristics, and from these I was able to create pairings. And in the first generation I created 3 pairings. The first one was a vestigial cross, male and female. And the second pairing was a male wild type with a female vestigial, and the third pairing was a female wild type with a male vestigial. And the reason I did that was to demonstrate that the characteristics they were inheriting were not sex. It was really important to do that, because if it turned out they were, that would have really affected the outcome of the results. So you can see here, after it was about 8 days, I had evidence of young, and it's difficult to make out on this image. There's actually some tracts in that food there. And there's the 3 original containers I had. Unfortunately, the Drosophila are very, very small, so they're quite difficult to actually photograph. But this demonstrated that I obviously sexed them correctly, because they have offspring. So what I had to do for the first generation was, after about 11 days, they emerged from the larval stage, and I could etherize the young. Now, I'd already removed the parents at this stage. I removed them as soon as the young reached the larval stage. I removed the parents because they were no longer needed. And after I'd etherized all of the young, I was able to actually examine them and record whether they had vestigial wings or wild-type wings. And from these young, the F1 generation, I then drew a second generation. And what I did was I took 2 male and 2 female from the vestigial cross, container A, and separate them into 4 other containers and introduce a male wild, 2 male wild, one from each of containers B and C, and 2 female wild from each container B and C, and create a new generation. And the reason I used the vestigial cross from the young from that particular cross is because I knew now that they had pure recessive genotype. So once again, I left these containers for approximately 11 days. They presented young again, and then it was a case of removing the parents whilst the offspring were in the larval stage. And then once the offspring had hatched and were actually developed flies, removing them again, etherizing them, and once again recording whether they had vestigial wings or wild-type wings. And you can see here some really good photos actually. You can see the larval stage of the software on the side of the container. And these are the raw results I came up with. top graph actually shows the F1 generation. So container A is a vestigial cross. Containers B and C are the vestigial and wild type cross. And then containers D to G are all vestigial wild type crosses. Now, these raw numbers on their own don't actually need a breakdown. So in order to compare them to members' research, I had to take the expected ratios that I should have had, which in the case of container A should have been 100% vestigial weight. And in the case of containers B, C, D, E, F, and G should have been a 3-to-1 ratio of wild type to every vestigial weight young. So I put these into a new table. And I applied a formula called chi-squared, which I know not everyone likes maths, but it's a pretty straightforward equation to calculate this. So what you can see there is the first column is the total number of young from each container. The second column is the observed wild/vestigial, and that's what actually existed. The third column along is the expected ratio that I should have had of wild to vestigial. And then I transformed this into a number, which is what I've got in the furthest columns of right here. Now, containers A to G were actually dead on what should have been expected. So it's a fairly straightforward formula, and you can see here how the result comes out as 0. But containers D and E were slightly off. So I had to minus the observed from the expected, and then divide that by the expected value, and then add the two values together for the two phenotypes, wild and vestigial, and that gave me a new number. But to get chi-squared for the overall results, I then had to come up with a mean number, which you can see is the bottom formula here, which gave me a final result of 0.0233. And when I applied this to the chi-squared table, it came back with a probability of 0.05 or less, which essentially meant the hypothesis could be accepted. Mendelian inheritance does provide a means to actually predict inheritance in dihydral plants. So why is this important? Well, the first application that comes to mind is actually in farming and agriculture. So these are two examples of what's called selective breeding. What we have here is a Belgian blue cow, which actually has a recessive mutation. It has a faulty gene that regulates myostatin, which means it has overdeveloped muscles. So it has a greater volume of much leaner meat. Now, if this were left just to be bred out, you have very small numbers of these cows because it is a recessive characteristic. However, because of selective breeding, there are entire herds of them, and they're separated out from the main wild type population of cows to preserve this particular desired characteristic. And the second is actually common corn, which is itself the result of selective breeding. And you can see here what it looks like in its natural state. And through selective breeding, what we've got in the end, which obviously we're all familiar with what corn looks like. Now, it's really important to note that nature isn't completely random. And this is why I showed the images of the nitrogenous-based pets before. Because these control every single possible characteristic. It comes down to DNA. Anything that you want to preserve or that you want to improve upon, you can isolate it and you can pass it on to future generations of any plant or any animal. And this is really important for increasing crop yields or coming up with plants that are slightly more durable in slightly off weather conditions. And what you'll get is much more successful plants because it's not left out of random chance. You can actually track what's been inherited and you can actually have a really well-targeted crop and animal and herds of animals. But there are also medical applications. And we're still very much exploring the human genome, but our understanding of it is actually increasing very good. And you can see on the left-hand side here, this is showing inheritance of Huntington's disease, which actually was identified through inheritance studies, obviously very similar to the one I did with using humans, and presumably not euthanizing them, showed that it's actually a genetically inherited condition, rather than something that's caused by exposure to a particular kind of environment. The same thing to be found to be true for osteoporosis, which is down to LRM4, a particular gene which causes faulty bone density. So once we can identify these genetic disorders through things like inheritance studies, and then compare those inheritance studies to sequenced DNA, which is something we are are capable of doing now, we can identify illnesses that have genetic causes, and we can locate these specific mutant genes that are causing these, which actually leads on to, once the faulty genes have been located, you can actually start treatment earlier. And over time, we're actually developing gene therapy, which hopefully will provide a cure altogether. And you will get to go to lunch shortly and stretch your legs. But before you do, does anyone have any questions they'd like to ask?
What's the actual difference between the two wind types and how easy is it to distinguish?
It's extremely easy. You can see the two images here are what's called wild type Drosophila. I mean, first of all, they can fly, and they do. They are an absolute pain in the neck to try and get these right and actually track them. One of the images I've got, I think it's one of the first ones. You can't make it out here, but when you're looking at it on that screen there, these are the vestigial Drosophila. You can just like out, yeah, if that's okay, because a lot of these images, they don't show up like this. There you go. You can see much, much smaller wings coming out the side there. They're extremely underdeveloped compared to the images you saw before, where the wings are almost full length of the body. And these guys are incapable of flight. They're actually much, much more docile. And you can tell usually by looking at them even without a microscope which ones are vestigial and which ones are a wild type. There are a few characteristics that Drosophila actually have. There's also body colour and eye colour, which are all dihybrid inheritance as well. I'd love to have studied more, but the problem with Drosophila is that the moment you start studying them, you're very much on their timetable. And Jolene and Sue were both brilliant in that they let me miss a couple of lectures to track these inheritance. But the moment they leave the larval stage, they become sexually mature within a matter of hours. So you've got to drop what you're doing and deal with them. And the lab techs were actually brilliant. They gave me when I came in at one point over the whole day to separate them out. Because obviously if they become sexually mature and they start mating before you can separate them, you've got to scrap the experiment and start over completely. So really it came down to practicality. But if I was to repeat the experiment, I'd like to carry on for a third, maybe a fourth generation and actually track one of the other characteristics